Well, you probably heard the expression, don't put all your eggs in one basket. I suppose it came of age during the time when most of us were agricultural, were farmers, and we collected eggs. So we all understood what that was about. You don't want to be walking down the path with your basket of eggs, the whole entire collection of eggs that's going to get you through the day or the week, and then trip and then break them all. You need to make sure that you have them in at least 2 baskets so some of your eggs get there. Well, that actually also applies throughout life. And according to Professor G, Dr. Gavin Giovannoni, we've been doing a little bit too much focusing on B cells lately instead of on T cells. Well, hello, and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam. I've been living with multiple sclerosis for 40 years, and I have been following with great interest all of the developments in MS, in understanding the causes and possible cures of multiple sclerosis, as I'm sure you all have too. A lot of the things that we look at together are actually fairly easy to follow. Even though they're scientific, they're pretty simple, even if you don't have a degree in science. But I have to warn you, today's is a little bit more technical. There's a lot of terminology that maybe we're not terribly familiar with. But I think we'll understand the bottom line when we get there. So let's go ahead. We'll take a look at a recent MS Selfie article from Professor G, and the article to which he refers. We'll take a quick look at that too. We'll try to see if we can't make some sense out of what this is all about. I saw that Professor G had posted something in MS Selfie on October 27th of 2024. Can we continue to ignore the evidence that T cells are fundamental to MS? Is it not time to take off the B cell blinkers? Okay, so we've been talking a lot about B cells lately and the possible damage that they cause to folks with multiple sclerosis. I think what Professor G's concern is that we're putting all our eggs in one basket again, and we're looking at one type of cell instead of the other, and that might be to the detriment of coming up with the real answer. So let's go and see what he has to say here. He writes, I have had many debates about whether or not MS is a T cell or B cell mediated disease. The emergence of B cell depleting anti-CD20 and B cell inhibiting BTKIs therapies as a treatment for MS means the pendulum has swung into the B cell camp. However, this ignores the solid circumstantial data that puts T cells as the orchestrator of MS pathology. Surely finding mainly CD8 plus T cells in MS lesions is telling us something? CD8 plus effector cells are usually involved with viral infections and are one clue I use to support the viral hypothesis of MS. I suspect B cells may be downstream of the T cell, and B cells may simply be involved as antigen-presenting cells, or dare I say, as the source of EBV, which is driving MS disease activity. And as we know, he's looking at EBV as the cause of multiple sclerosis, and that if we could eliminate EBV, we'd eliminate multiple sclerosis. Lovely thought. Not as easy to do, and he's the first one to admit that, not as easy to do as we would like, but I have done a video on that recently, which I'll link below, and you can listen to the latest thinking on that. I was therefore very excited to see the study below that examined CD8 plus T cells in people with multiple sclerosis and their identical twins to identify immunological and metabolic changes associated with the disease. In short, the findings, and I find this very useful that he's doing key findings for us. He says, first of all, that CD8 plus T cells in persons with MS and their twins with subclinical neuroinflammation, SCNI, or arguably asymptomatic MS, showed signs of being more activated, pro-inflammatory, and migratory than healthy individuals. These changes were evident in CD8 plus T cells from the blood and cerebrospinal fluid CSF. And then the second point is that specific gene expression patterns in CD8 plus T cells, including several markers, CD827, CD869, COTL1, and ANXA5, are associated with increased T cell activation capacity. And the third point that he derived from the paper was using an analysis gene set enrichment analysis, or GSEA, they revealed that pathways related to interferon gamma response, in other words, IL-2/STATS signaling and oxidative phosphorylation, were enriched in both SCNI and MS groups. The next point is that CD8 plus T cells from MS-affected individuals showed higher expression of genes involved in proteasomal degradation and HLA class II antigen presentation than those with SCNI. And the next point is that the study identified several immunological modules linked to increased activation, antigen presentation, central nervous system migration, and production of effector molecules in MS disease-associated CD8 plus T cells. Next, he says, metabolic module analyses revealed upregulated energy metabolism, specifically oxidative phosphorylation, in SCNI and MS groups. This was evidenced by the high expression of genes involved in glucose intake, or SLC2A3, glycolysis or glucose metabolism, GAPDH, LDHB, the citric acid cycle, MDH2, IDH2, and the mitochondrial respiratory complex 1, NDUFA13. And they confirmed the immunological and metabolic alterations observed in the twin study in an independent validation cohort of untreated patients with relapsing remitting MS. And the last key finding was that single-cell analysis of brain tissue from MS patients showed that T cells in MS lesions also express key elements of the immunological and metabolic modules supporting the sustained activation of CD8 plus T cells in these areas. In summary, he says, this study identified specific immunological and metabolic alterations in CD8 plus T cells that are associated with both the prodromal, which means the symptoms you get before you're actually diagnosed with MS, or asymptomatic and manifest stages of MS. These findings suggest that CD8 plus T cells play a key role in MS pathogenesis and highlight potential targets for diagnostic and therapeutic approaches. You may have found the earlier section pretty technical and complicated. I know I kind of did. But let's hope that here it's a little more clear, because he's going to talk about the implications of these findings for clinical practice. He says that the findings offer several potential implications for clinical practice, highlighting potential avenues for diagnosing, monitoring, and treatment of the disease. And the first one is that early diagnosis and monitoring is important. The study identified immunological and metabolic alterations in CD8 plus T cells present even in individuals with SCNI, suggesting that these changes occur early in the prodromal or asymptomatic stages of MS. So it sounds like it's an important biomarker. This raises the possibility of using these alterations as biomarkers. Aha, look, I'm actually getting good at this. For early diagnosis and monitoring disease progression, potentially allowing for earlier intervention. This could involve monitoring the expression of specific genes or proteins in CD8 plus T cells, or assessing the metabolic activity of these cells. And then he goes on to list some therapeutic targets. Identifying specific immunological and metabolic pathways dysregulated in MS-associated CD8 plus T cells opens up potential therapeutic avenues. And then he's going to go on to list a bunch of these. For instance, targeting the IL-2 signaling pathway, particularly IL-2RB, which is upregulated in SENI, could be beneficial in the early stages of the disease. And then the second point is that inhibiting CXCR3, a chemokine receptor involved in T-cell migration to the CNS, could be another approach to modulate T-cell trafficking and reduce neuroinflammation. Next, he says targeting CD6, which mediates interactions between T-cells and antigen-presenting cells and is highly expressed in MS-associated CD8 plus T-cells, might also be explored. The next point is that the study's findings also suggest that JAK inhibitors, already used in other autoimmune diseases, could hold promise for treating MS due to JKA3's involvement in T-cell activation. And then finally, the potential of targeting metabolic pathways, particularly oxidative phosphorylation, for therapeutic intervention is also highlighted. And then the last implication he talks about is understanding disease mechanisms. The in-depth characterization of CD8 plus T-cell alterations in MS provides valuable insights into the underlying disease mechanisms. The study demonstrates the critical role of CD8 plus T-cells, in driving inflammation. It highlights their complex interplay with other immune cells, such as dendritic cells and macrophages, by releasing chemokines like CCL5 and FLT3L. This more profound understanding of disease pathogenesis can guide the development of more targeted and effective treatments. And in conclusion, he says, overall, the study underscores the importance of CD8 plus T cells in MS pathogenesis and offers a rich source of potential targets for developing novel diagnostic and therapeutic strategies. Further research is necessary to translate these findings into clinical applications, link them to EBV biology, and determine the most effective ways to manipulate these pathways as a treatment for multiple sclerosis. This is the paper that he's referring to. It's in the journal Science Immunology. It was published on 27 September of 2024. Twin study identifies early immunological and metabolic dysregulation of CD8 plus T cells in multiple sclerosis. And I looked up the affiliations of the many authors on this paper, and they do, by and large, seem to be coming from Munich, Germany, a university there. And just to take a quick look at the editor's summary, which I appreciate they put up here because it's a lot easier to see what's going on than have to wade through all the technical methodology and experimental this and that, because, most of us just don't really understand most of this. It says that multiple sclerosis is an inflammatory disease of the nervous system in which CD8 T cells are present in MS lesions, but their role in disease progression remains unclear. Kavaka et al. Analyzed CD8 T cells from the blood and cerebrospinal fluid of monozygotic twins, so that would be identical twins, where one twin had MS and the other showed no signs or subclinical signs of neuroinflammation, so that's SENI. CD8 T cells from individuals with MS and co-twins with SENI exhibited pro-inflammatory, immunological, and metabolic features consistent with enhanced activation and migration. These findings were validated in a separate cohort of individuals with MS, providing insights into the role of CD8 T cells in MS progression. Well, okay, so I guess I get this a little bit better than I did, maybe, that if nothing else, the presence of the T cells, the CD8 plus T cells, makes a really good biomarker to let you know that there's disease activity going on. They do seem to pertain a great deal to neuroinflammation, And there's bound to be some other things that are related to the T cells that are quite destructive for those of us with MS. So, like eliminating the B cells, as we're trying to do with the BTK inhibitors, we also need to be looking at how can we control the propagation of T cells, at least the ones that spell bad news for those of us with multiple sclerosis. And I mean, you're quickly getting in over my head, I have to say. There's an awful lot that goes on. I don't know much about microbiology or cellular biology beyond what I read, so I'm not going to be able to help you wade through all this. But one thing that we do know is that reputable scientists are looking at all sides of this, and that's probably the best thing we can be doing, is just trying to keep up with what they say and what they're thinking. Sooner or later, maybe it'll all click and make sense, I don't know. But I think the more I try to follow along with what's happening, the better off I am anyway. I feel that slowly but surely things are beginning to make a little more sense to me. All I know is that MS is an incredibly complicated disease. It's nowhere near as simple as I thought when I was first diagnosed years and years ago. And I'm sure that you're feeling the frustrations that come from that complexity, because there's no simple answer. When we go to the doctor, they don't just give you a pill or a shot or something, tell you don't eat this food or do that exercise. There's nothing that simple, and there probably never will be. I'd like to believe that if we get that shot for Epstein-Barr virus, that at least the people who don't already have MS will never get it. But as Professor G said in the last video I made on that subject, we don't know. It's going to take years and years, and probably longer than most of us are going to be alive to even find out whether that would be true or not. You just have to wait it out. MS is a long-term disease. It shows up at whenever it feels like. It takes the time it takes to get going. It can be kind of tricky to even diagnose, even nowadays, when they tend to be a lot faster about it because their diagnostic tools are better. So we just have to be patient. I guess that's the bottom line. It is frustrating, incredibly frustrating, but I don't see that we have any choice but to be patient. At least we know that it's being worked on. It's not for lack of effort that answers aren't coming. And we have to take heart in that, at least. But until I see you in my next video, the thing to do, right, is to take really good care of yourself so that we can be around for as long as possible and take advantage of whatever comes down the pike, because you just never know. I'll see you again in my next video.